Hi students, today we are going to learn about physiological anatomy, neuronal circuits and functions of basal ganglia. First coming to the embryological development of basal ganglia, this is the primary division of the neural tube, which is divided into rosencephalon, mesencephalon and rhombencephalon, and it is divided further into secondary subdivisions. They are telencephalon, diencephalon from the prosencephalon, mesencephalon from the mesencephalon, metencephalon and myelencephalon from the rhombencephalon. So, three primary vesicles will be developed into 5 secondary vesicles. Then, what are the final segments in a human adult? So, from the telencephalon, on each side of the brain, it forms the cerebral cortices, caudate, putamen, globus pallidus and ventral pallidum. This diencephalon forms thalamus, subthalamus, epithalamus, hypothalamus and subthalamic nuclei. This mesencephalon forms midbrain, substantia nigra, pars compacta and substantia nigra, pars reticulata. Metencephalon forms pons and cerebellum and myelencephalon forms medulla. So here we are discussing about the basal ganglia. All these highlighted ones are included under basal ganglia. So they are formed from telencephalon, diencephalon, which are parts of prosencephalon, and from mesencephalon. The basal ganglia are subcortical masses of nuclear masses. They are coming under extrapyramidal system. Physiologically, they are divided into three parts. Corpus striatum, which includes caudate nucleus and lenticular nucleus. This lenticular nucleus includes putamen and globus pallidus. So, corpus striatum is having caudate nucleus and lenticular nucleus. Then, second part is subthalamic nucleus. The third part is substantia nigra. So, physiologically it is divided into three parts: corpus striatum, subthalamic nucleus and substantia nigra. Then phylogenetically and functionally, it is divided into neostriatum or striatum, which is further divided into dorsal striatum and ventral striatum. The dorsal striatum consists of caudate and putamen. Okay, this caudate and putamen, and ventral striatum is having nucleus accumbens and olfactory tubercle. Then coming to paleostriatum or pallidum or globus pallidus. It is further divided into dorsal and ventral pallidum. Okay, so physiological classification and phylogenetically and functional classification is given here. Physiologically into three parts, phylogenetically and functionally into two parts, neostriatum and paleostriatum. See here, these are the basal ganglia which are shown in red, and the related structures are shown in blue. Okay, this is caudate nucleus. This is globus pallidus. This is putamen. Okay, and this is subthalamic nucleus, and this is nucleus accumbens. This is olfactory tubercle. This is ventral pallidum. The structures associated with are amygdala, thalamus, and here we have substantia nigra, which is a part of basal ganglia, and the related structure is ventral tegmental area. Here you can see thalami, and here you can see basal ganglia. Okay, on either side. This is putamen, this is caudate nucleus. Okay, this is globus pallidus, this is amygdala. Coming to physiological anatomy of basal ganglia. In terms of anatomy, the basal ganglia are divided into 4 distinct structures depending on how superior or rostral they are. In other words, depending on how close to the top of the head they are. Two of them, the striatum and the pallidum, they are relatively large, and other two, substantia nigra and subthalamic nucleus, are smaller. Here it is showing coronal slices of the human brain, which is showing basal ganglia. So anteriorly you have striatum and globus pallidus. Striatum and globus pallidus anteriorly. Okay. And posteriorly you have subthalamic nucleus and substantia nigra. Coming to neostriatum or striatum. So here we are discussing the phylogenetical and functional classification. Neostriatum is also called as striatum. What it includes? It includes dorsal striatum and ventral striatum. The dorsal striatum includes caudate nucleus and putamen. This is caudate nucleus, head, body and tail. This is caudate nucleus and putamen. These are coming under dorsal striatum. Okay. And ventral striatum consists of nucleus accumbens and olfactory tubercle, somewhere here. Okay, this is nucleus accumbens, and here you have olfactory tubercle. So neostriatum or striatum is divided into dorsal striatum and ventral striatum. The dorsal or neostriatum is divided into caudate and putamen by internal capsule. So the neostriatum, okay, that is the dorsal striatum, is divided into two parts, that is caudate nucleus and putamen. So in between you have internal capsule. The globus pallidus plus putamen is together called lentiform nucleus. Okay, globus pallidus and putamen. So globus pallidus is a part of paleostriatum or pallidum. So here you should notice that a part of the neostriatum, that is putamen, and a part of the paleostriatum, that is globus pallidus, together consists of lentiform nucleus. Okay. So it is a combination of neostriatum and paleostriatum. When the ventral striatum is composed of nucleus accumbens and olfactory tubercle, as we have discussed. See here, these are the parts. Okay. Here you have globus pallidus externus and internus. Okay. And you have putamen. Okay. Here you have internal capsule, and here you have caudate nucleus. So internal capsule divides the caudate nucleus and putamen. And this putamen and globus pallidus together consists of lentiform nucleus. Okay. This is subthalamic nucleus. This is substantia nigra. Okay. Caudate nucleus will come like this: head, body, and tail. Okay. So caudate nucleus. So yeah, we are discussing about caudate nucleus. It is a comma-shaped band of grey matter. So caudate nucleus is comma-shaped band of grey matter. It has three parts: head, body, and tail. So this is the head of the caudate nucleus. Okay, and this is the body of the caudate nucleus, and this is the tail of the caudate nucleus. Okay, you see here the red one is a caudate nucleus. Coming to putamen, this is putamen, which is again they have shown in the red color. This is putamen. This is caudate now, and this is putamen. Caudate means having a tail or tail-like appendage. So it has a tail or tail-like appendage. This word, next coming to putamen, the word putamen is from Latin. Okay, referring to that which falls off in pruning, from putare, meaning to prune, to think or to consider. Okay, so putamen word taken from Latin means falls off in pruning. Okay, so they have considered it as it has fallen off when the brain is pruned. This is the word coming from putare. The striatum is composed mostly of medium spiny neurons. These are the medium spiny neurons. So the striatum. So striatum means what? We are talking about neostriatum. Okay, neostriatum or a striatum. So it has dorsal and ventral striatum. You already know. So we are talking about striatum or neostriatum. So here it is composed of medium spiny neurons. These are GABAergic neurons which project to the external globus pallidus and internal globus pallidus. External globus pallidus is located laterally and internal globus pallidus is located medially. Okay, and these are also projecting to the substantia nigra pars reticulata. Okay, so these GABAergic neurons will project to three parts. What are those three parts? Globus pallidus externus and internus, as well as substantia nigra pars reticulata. So you have in the striatum you have dorsal and ventral striatum. No, so dorsal striatum, which is having these two parts, that is caudate and putamen. What are the connections to this dorsal striatum? Here you can see this is the striatum. Okay, which is having caudate nucleus and putamen. So the dorsal striatum receives glutamatergic inputs from the cortex. Okay, it is receiving glutamatergic inputs. That is, glutamine is the neurotransmitter. It is released from the cortical neurons. And dopaminergic inputs from the substantia nigra pars compacta. This is substantia nigra, which is again divided into two parts. Substantia nigra pars reticulata and substantia nigra pars compacta. So from the pars compacta, it is receiving dopaminergic inputs. Okay, so it is receiving two inputs. What are those inputs? From the cortex glutamate as a neurotransmitter, glutamatergic input, and from the pars compacta, dopamine, that is dopaminergic input. The dorsal striatum is considered to be involved in sensory motor activities. So it is important for performing sensory motor activities. Coming to the connections of the striatum, we have already seen it receives afferent input from the cerebral cortex and substantia nigra pars compacta, and also from the intralaminar thalamic nuclei. And also from the intralaminar thalamic nuclei. Intralaminar thalamic nuclei means the nuclei which are present in the Y-shaped structure, Y-shaped white matter in the thalamus. Then efferents will go to globus pallidus and substantia nigra. That is, efferents will go to where? So in the previous diagram it has shown efferents will go to globus pallidus externus and internus. Okay, and so direct connections we are talking. Okay, so from the striatum the efferents will go to globus pallidus externus and internus, and to the substantia nigra pars reticularis. Pars reticularis. Then coming to the connections of the striatum. So what are those connections to the striatum? What are the afferent fibers and what are the efferent fibers? So what are the afferent fibers? You have already studied. Corticostriatal. from cortex to the striatum. Okay, striatum means caudate and putamen, neostriatum. Cortex to striatum, that is corticostriatal. It is releasing glutamate as a neurotransmitter. Then thalamostriatal. Thalamostriatal, from thalamus to the striatum. That is, here you can see from the intralaminar nuclei of the thalamus, the input is coming. Third one is the nigrostriatal. Okay, third one is the nigrostriatal. From the substantia nigra pars compacta, the input is coming to the striatum. It is releasing dopamine. Then it is also receiving fibers from the brainstem. Brainstem striatal. So these four are the inputs, that is afferent fibers to the striatum. Now coming to the efferent fibers from the striatum. Striatopallidal. Striatopallidal means to the globus pallidus. It is projecting its fibers to globus pallidus externus and globus pallidus internus. And also it sends its fibers to the substantia nigra pars reticularis. Okay, substantia nigra pars reticularis. Then coming to the types of cells in the striatum. What are the types of cells which are present in the striatum? They are medium spiny neurons which are releasing GABA, that is an inhibitory neurotransmitter. Okay, D1 and D2 types has DR1 and DR2 receptors respectively. So the cells in the striatum are releasing GABA, which are medium spiny neurons. So these GABA is inhibitory. Okay, so this can act on the D1 and D2 types, and this D1 has a DR1 receptors and D2 is having DR2 receptors respectively. Next thing is, it is having a spiny cholinergic interneurons. Themselves are affected by dopamine through DR5. So it is also having second kind of cells. They are aspiny. So this is spiny neurons. These are aspiny. They are releasing cholinergic acetylcholine, cholinergic interneurons. Okay, they are affected by dopamine through DR5 receptors. And there are many types of GABAergic interneurons. These are not distributed evenly. Okay, so there are many kinds of neurons. There are GABAergic interneurons, medium spiny neurons which are releasing GABA, aspiny cholinergic interneurons which are releasing acetylcholine. Next, coming to functions of the caudate nucleus. So what are the functions? It is performing motor functions and cognitive functions. Okay, so we have discussed what are the classifications of the basal ganglia, and that we have taken functional and phylogentical classification. Okay, so phylogenetically it is divided into neo and paleostriatum. In that we are discussing neostriatum or striatum, that is caudate nucleus and putamen. Okay, we have seen afferent and efferent connections to that. And now we came to the functions of the caudate nucleus, which is performing motor and cognitive functions. So the motor functions are spatial mnemonic processing. The caudate nucleus integrates spatial information with motor behavior formulation. So it will integrate spatial information with the motor behavior. Then it performs directed movements. It contributes importantly to body and limbs posture and the speed and accuracy of directed movements. Okay? So it will control the speed and accuracy of the directed movements. So two motor functions. spatial mnemonic processing, and directed movements controlling. Then coming to cognitive functions. So it will perform all these functions. What are those? Goal directed action. A strong relationship between the caudate and the cortical areas associated with executive functioning. So it will perform goal directed action, and it is also helpful for working memory and learning, mainly correct categorization, and it is responsible for deep slow wave sleep, and is also responsible for emotion and language. So it plays a part in all these functions. Threshold control. Regulatory role by measuring the general activity of cerebral cortex and controlling the threshold potential. Okay, so it will perform its a regulatory role while performing any action. It will control the threshold potential in the cerebral cortex. Then we came to putamen. What are the functions of the putamen? These are it will perform motor skills, and also it forms a part in learning. The motor skills are, it will play its action in the motor planning, learning, execution, motor preparation, specifying amplitudes of movement and movement sequences, and plays a role in the selection of movement and the automatic performance of previously learned movements. Okay, so it will perform all these motor activities, and it also plays a role in learning. It is important for reinforcement and implicit learning. Okay, so while we studying the memory and learning methods, we will learn about what is implicit learning, what is explicit learning, and etc. Okay, so it will, the putamen will form its role in the reinforcement and implicit learning, and it is important for category learning. And head circuit, it forms a head circuit which involves the putamen and the insula. So along with the insula, it forms the head circuit. Now we came to paleostriatum. In that you have globus pallidus, internus and externus. The globus pallidus is a Latin word, means pale globe. Here the globus pallidus is shown in the red within the brain. Okay. So mainly the pallidum consists of dorsal pallidum and a small ventral extension which is called ventral pallidum. So mainly we are concentrating about dorsal structures. Globus pallidus internus and globus pallidus externus. They are separated by medial medullary lamina. Okay. So these two are separated by medial medullary lamina. They are having GABAergic neurons. So they will exhibit inhibitory effects on their target organs. And these also will participate in distinct neuronal circuits. Okay. The globus pallidus externus receives input mainly from the striatum and projects to the subthalamic nucleus. Okay. So the globus pallidus externus receives input from the striatum, and it mainly projects to the subthalamic nucleus. Whereas the globus pallidus internus receives signals from the striatum via the direct and indirect pathways. So what is the direct pathway? From striatum to globus pallidus internus. What is the indirect pathway? From the striatum to the globus pallidus externus, then globus pallidus internus. Okay. Then where it will project globus pallidus internus to the thalamus. Okay. So globus pallidus externus to the subthalamic nucleus. Globus pallidus internus to the thalamus. It will project. Then coming to the disinhibition principle. Disinhibition principle. The pallidal neurons, these neurons operate using a disinhibition principle. These neurons fire at steady high rates in the absence of input, and signals from the striatum cause them to pause or reduce their rate of firing. So what is meant by disinhibition principle? These parallel neurons will fire at a steady high rates if the input is not present. And if the input is present from the striatum, okay, the striatum will release GABA on these neurons and it will decrease the rate of firing. Because parallel neurons themselves have inhibitory effects on their targets, the net effect of striatal input to the pallidum is a reduction of the tonic inhibition exerted by pallidal cells on their targets, that is disinhibition, with an increased rate of firing in the targets. So what you understand by all these things? So parallel neurons will fire at a steady high rates to their concerned output organs, okay, and they are releasing GABA, so they are under inhibitory condition. That is, the targets of the parallel neurons are under inhibitory condition. Now when the striatum is stimulated, because the striatum is releasing GABA into the pallidum, because the GABA inhibits the pallidal neurons, then what happens? The targets of the pallidal neurons are disinhibited, so they will increase their rate of firing. Okay, that is the disinhibition principle. Now coming to the connections of the globus pallidus. Okay. So what are the connections of the globus pallidus? This is the internal and external globus pallidus. So what are the afferent connections? You know, that is from the striatum, direct and indirect pathways to the internal and direct pathway to the external. Striatopallidal fibers. Striatopallidal fibers. And from the subthalamic nucleus. From the subthalamic nucleus. Subthalamic nucleus will give its afferents to the globus pallidus internus. Globus pallidus internus. Okay, from the subthalamic nucleus, they will pass laterally through the internal capsule as subthalamic fasciculus and terminate in both segments of globus pallidus, but more in the medial segment, means mainly they will go to globus pallidus internus. Okay, so afferents from striatum and subthalamic nucleus. Then what are the efferent connections? From where it will go? Globus pallidus. So it will go to the globus pallidus externus. You know, go to subthalamic nucleus. That is, lateral means externus. It will go to subthalamic nucleus. The medial one, that is internal one, will go to thalamus, mainly to the motor areas. What are those motor areas? Ventro-anterior and ventromedial nucleus and centromedian nucleus. Mainly to motor areas. Okay, the efferents will go. Okay, from the internus to the thalamus. So in the previous slide, I said ventro-anterior, ventromedial nucleus. That is wrong. You concentrate on here. Okay. So it is projected to the motor nuclei, that is ventro-anterior, ventrolateral, and to the centromedian nucleus. Okay. Centromedian nucleus is a non-specific nucleus. So this is the afferent and efferent connections of the globus pallidus. So see here, all this circuitry is on the same side of the brain, that is, it is uncrossed. Thus the basal ganglia affect function mediated by the ipsilateral motor cortex. Okay, so the basal ganglia will affect the function mediated by the ipsilateral motor cortex. Since the motor cortex is controlling movements of the contralateral body, the basal ganglia affects the movements of the contralateral body. You understood this? Okay. So here they have given connections. They have given connections. This is the basal ganglial parts. I think you have understood all these things. Coming to the loops. primary feedback loop. Okay, or cortex, basal ganglia, motor cortex circuit. So what are the loops? Primary feedback loop, or it can be called as cortex, basal ganglia and motor cortex circuit. Here you see cerebral cortex. From all parts of the cerebral cortex, the information is coming to the striatum. From the striatum, it is going to the globus pallidus internus, okay, mainly, and to the, so in the primary loop you have all these things. So in the primary loop you have globus pallidus internus and substantia nigra pars reticulata or reticularis. So why it is shown in the dotted line and why it is showing in the continuous line? So the continuous line will inform that it is exciting this part. So dotted line is showing this part will inhibit this part. Okay. So the cerebral cortex is exciting the striatum because the striatum is having GABA neurons. It is acting on the globus pallidus internus and substantia nigra pars reticularis. It is inhibiting this part. Actually, this is inhibiting the thalamus. This is inhibiting the thalamus. Okay. But the thalamus is activating supplementary motor cortex and striatum. So this is the primary feedback loop. So if a movement has to be occurred, then what will happen? This cortex will stimulate the striatum. The striatum will inhibit this internus and the reticularis. Then what will happen? Because of the disinhibition principle, this effect will be lost. Then the thalamus will fire and it will send its impulses to the motor cortex, that is supplementary motor cortex, and also to the striatum. Okay. So this is the primary feedback loop or cortex, basal ganglia, motor cortex circuit. Here in this loop, you have this loop, caudate loop and putamen loop. Okay. So this caudate loop is receiving actually inputs, that is afferents from the supplementary motor cortex and motor sensory area. This will stimulate the caudate nucleus. Okay. Then this will stimulate the globus pallidus internus, the caudal part, and substantia nigra pars reticularis caudal part. So the caudal parts, they are stimulated by caudate nucleus. And this caudal part of the GPI will stimulate, actually it will inhibit the VLO, that is ventrolateral nucleus pars oralis. Ventrolateral nucleus pars oralis. This reticularis will inhibit the, actually it is inhibiting the VLM, that is ventrolateral nucleus pars medialis. Okay. So these are the thalamic parts which are get affected by the globus pallidus internus and substantia nigra. Whereas here, if you see this putamen loop, the putamen nucleus is receiving its afferent fibers from the frontal association area, and this will send its impulses to the rostral part of the globus pallidus internus and substantia nigra pars reticularis. Whereas these are influencing ventro-anterior nucleus parvocellular and ventro-anterior nucleus magnocellular. Okay, these are the parts of the thalamus. So these two loops will be combined in the previous slide. So caudal part is connected to caudate nucleus. C you can remember like that. And these are connected to these types of nuclei in the thalamus. And here you should remember that there is one term called hyperdirect pathway. So far you have seen direct and indirect pathways. You have hyperdirect pathway, means the cortex is connected to subthalamic nucleus directly. Okay. So the cortex will stimulate the subthalamic nucleus and it will stimulate the globus pallidus internus and reticularis, which is inhibiting the thalamus. So it is called hyperdirect pathway. Now we are going to see indirect pathway, which is going to involve substantia nigra pars compacta. What is that? So here this Diagram is showing indirect pathway of feedback loop involving substantia nigra pars compacta. Okay, this is actually comes under modulation of the basal ganglia. So two neuronal circuits will modify the function of the basal ganglia. Those are nigrostriatal pathway and thalamostriatal pathway. So coming to the nigrostriatal pathway, this pathway projects from the substantia nigra pars compacta to the striatum. Okay, and this utilizes the neurotransmitter dopamine. This pathway, as you already know, that it has a modulatory effect on the basal ganglia. And what this will do, okay, it will facilitate the motor loop in two ways. That is, it excites the direct pathway and it inhibits the indirect pathway. Okay, so this pathway is very much important. So to excite the direct pathway and to inhibit the indirect pathway. Okay, so we have seen direct pathway and indirect pathway earlier in the earlier slides. We have noticed here that there are two types of dopamine receptors in the striatum. Those are D1 receptors and D2 receptors. So if you stimulate the D1 receptors, results with the excitation of the neuron, while the D2 receptors, if they are stimulated, they will inhibit the neuron. D1 receptors are found on the striatal neurons, okay, that give rise to the direct pathway. So D1 for direct pathway. So D1 are important for a direct pathway. So our SNPC is stimulating direct pathway, okay, and D2 receptors are involved in the indirect pathway. So it is inhibiting the indirect pathway. So what is the final effect? That is stimulation of the direct pathway, and at the same time it will also inhibit the indirect pathway. Okay, so it is also known that the activity of the neurons within the substantia nigra pars compacta is related to the reward system and behavioral stimuli, and it is also important for motor learning. Okay, and the other one which is modulating the activity of the basal ganglia is thalamostriatal pathway. That is, from the intralaminar nuclei of the thalamus, the fibers will arise and they will terminate on the striatum. Okay, so these two will modulate the function of the basal ganglia. So why these are like this? Because the basal ganglia are nuclei. Actually, these are not ganglia, these are nuclei. Okay, they are heavily interconnected and plays an important role in the motor planning and modulation. Okay, so with the help of these three pathways, that is direct, indirect and hyperdirect pathway, they will do the motor movement. Okay, so we have seen the direct pathway, we have seen the indirect pathway which is involved in the subthalamic nucleus. So disinhibition principle you should remember, and we have seen the indirect pathway which is having subthalamic nucleus. We have already discussed that, and the other indirect pathway which is involving SNPC, which is important for modulating the action of the basal ganglia. So it will stimulate the direct pathway and it will inhibit the indirect pathway. And the thalamus is also projecting onto the striatum and it will also modulate the action of the basal ganglia. So here you can see this is direct pathway, this is indirect pathway. So direct pathway will facilitate the movements and indirect pathway, it is inhibiting the unwanted movement. Okay, so it is inhibiting the unwanted movement. So in the direct pathway you have two pathways. You have already seen that caudate loop and putamen loop. The indirect pathway you have subthalamic nucleus, and the indirect pathway which is controlling the modulation of the basal ganglia are involving substantia nigra, pars compacta, and from the thalamus also, thalamostriatal fibers. So you have understood this. So ultimately the movement will be facilitated and the unwanted movement will be inhibited. Now coming to the ventral striatum. Okay, so we have already seen what is this ventral striatum or ventral pallidum. There is a small ventral extension from the dorsal pallidum, that is ventral pallidum or ventral striatum. Okay, so what nuclei it is composed of? See here, the ventral striatum is composed of nucleus accumbens and olfactory tubercle. Okay, these two will is called together called ventral striatum, and it has the similar output pathways to the globus pallidus while receiving input from other gain setting nuclei such as the ventral tegmental area. So it will receive from the ventral tegmental area and output to the globus pallidus. You can see here, this is the diagram showing nucleus accumbens. Okay, nucleus accumbens. The ventral striatum, actually it receives glutamatergic inputs from the limbic areas. Okay, so amygdala for example. Okay, so these are the limbic areas. So it receives inputs, that is glutamatergic input from the limbic areas, and it is a vital part of the circuitry for decision making and reward related behavior. Okay, so ventral system is important for decision making and reward related behavior because it is connected to the limbic system. See here, the dopaminergic inputs from ventral tegmental area will come through mesolimbic pathway. Okay, so this is ventral striatum. Okay, so ventral tegmental area, this is ventral tegmental area. Dopaminergic neurons will be projecting to the striatum, that is ventral striatum, from the ventral tegmental area. So you have noticed that it will receive its inputs, okay, from the ventral tegmental area via the mesolimbic pathway and from the limbic system. Okay, from the limbic system it is getting glutamatergic inputs. From the mesolimbic pathway it is getting dopaminergic inputs. Here a word about mesolimbic pathway. It is also called reward pathway. Okay, it is related to emotion regulation. You already know that the neurotransmitter which is released is dopamine. Okay, so these two areas are connected to limbic system and ventral tegmental area. So ventral tegmental area carries dopaminergic neurons via the mesolimbic pathway. It is important for regulation of the emotions. The pathway connects ventral tegmental area, which is present in the midbrain, to the ventral striatum of the basal ganglia, which is present in the forebrain. So there is a connection between the midbrain and forebrain. The release of dopamine from the mesolimbic pathway into the nucleus accumbens regulates incentive salience, that is motivation and desire for rewarding stimuli. Okay, and facilitates reinforcement and reward related motor function learning. So if you give any reward or any prize to a person, the person will do more work. Okay, so it is through mesolimbic pathway. So because of the release of the dopamine. Okay, and this function is called incentive salience, that is motivation and desire for rewarding stimuli. So they want to do more work and they want to get more rewards. And it may also play a role in subjective perception of pleasure. The dysregulation of the mesolimbic pathway and its output neurons in the nucleus accumbens plays a significant role in the development and maintenance of an addiction. So it is important for addiction. Addiction is mainly because of the dysregulation of the mesolimbic pathway. And it is also involved in the regulation of voluntary movement. Then coming to a pallidonigral pacemaker. The two pallidal nuclei and the two parts of the substantia nigra, pallidonigral, okay, they will constitute a high frequency autonomous pacemaker. So, pallidal nuclei internus and externus along with the substantia nigra, compacta and reticularis, they will constitute high frequency autonomous pacemaker. It is called pallidonigral pacemaker. Here some important points are there. The striatum 90% we have GABAergic neurons. They will project to the pallidum, and also dopaminergic axons from the pars compacta of the substantia nigra. You have already seen two types of receptors and two types of receptors are present in the striatum, which are influenced by substantia nigra pars compacta. Coming to the substantia nigra. Substantia nigra means black substance. It is present in the midbrain and it is a grey matter. It has two divisions, substantia nigra pars compacta and substantia nigra pars reticularis. Substantia nigra pars reticularis is working with the unison with the globus pallidus internus, and substantia nigra pars reticularis and globus pallidus internus complex inhibits the thalamus. Substantia nigra pars compacta, this however produces dopamine as a neurotransmitter which is involving in the balance of the striatal pathway. You have already seen. Okay. This substantia nigra pars compacta projects to the striatum and the portions of the cerebral cortex. Okay. And it releases dopamine which is involved in the fine motor control. Okay. The damage to this will cause to Parkinson disease and epilepsy. So in epilepsy, there will be decrease of this pathway 80%. Substantia nigra pars reticulata releases GABAergic neurons. The GABA as a neurotransmitter. Okay. They are fast spiking pacemakers. So substantia nigra pars reticularis is having fast spiking pacemakers, and it will generate the action potentials in the absence of the synaptic input. Okay. It will generate 68 hertz. Okay, in contrast to the dopaminergic neurons, they are stimulating below 8 hertz. That is afferents to the striatum. So you know that substantia nigra pars reticularis fights at the level of 68 hertz. Okay, and substantia nigra pars pars compacta can generate impulses at the rate of 8 hertz. Okay. This reticularis receives axons from the subthalamic nucleus and a dopaminergic innervation from the dopaminergic ensemble. Okay, you have already seen no subthalamic nucleus in the indirect pathway. Okay, so what is happening here is you already know that substantia nigra pars reticularis and globus pallidus, they will generate they will have pacemakers and they can generate fast spiking pacemaker is present, so they can generate this much impulses. Okay. This figure has shown location of the substantia nigra within the basal ganglia. So this is the substantia nigra on either side. This is a micrograph of neurons containing neuromelanin. So the arrows, they are pointing towards the neuromelanin. So this neuromelanin is present in the substantia nigra. It is studied in the rhesus monkey brain. Okay. This blue colored is the Nissl stain. So because of the presence of neuromelanin, substantia nigra is black in color. Then the pars reticulata is one of the two primary output nuclei of the basal ganglia system to the motor thalamus. Okay. The other output is the internal segment of the globus pallidus. So you can see that globus pallidus internus and reticularis, they will work together, and they are the primary output to the thalamus. This sends axons to the frontal cortex and oculomotor cortex also. And this is largely involved in orientation and control of eye movements, in stabilization of the gaze, and in saccades. So they will stabilize the control of, they will stabilize the gaze, and they will control the movements of the eyes. Pars reticulata sends inhibitory projections to the superior colliculus and inhibits the eye movement. And this inhibition is lifted, that is, the projecting neurons cease firing during saccades. So what you have learned? The pars reticulata is projecting to the thalamus, and it can also project to the frontal cortex and oculomotor cortex, and it will control the eye movements, and it stabilizes the gaze, especially in saccades. So how it is doing? This reticulata sends inhibitory projections to the superior colliculus, thus inhibits the eye movement. Okay, inhibits the eye movement. So if pars reticulata is damaged, this inhibition is not there to the superior colliculus. So the saccades will be appearing. Now coming to the body of Lewis or subthalamic nucleus. This is the only portion of the basal ganglia that produces an excitatory neurotransmitter called glutamate. Okay, so this is the uh, glutamate neurotransmitter which is released from the subthalamic nucleus. This is also called body of Lewis. The role of the subthalamic nucleus is to stimulate the substantia nigra pars reticularis and globus pallidus internus complex via the indirect pathway. You have already studied this subthalamic nucleus is involved in the indirect pathway. Okay. The subthalamic nucleus receives inhibitory input from the external part of the globus pallidus and sends excitatory input to the globus pallidus internus. So what you have seen in the indirect pathway, this subthalamic nucleus is stimulating this complex. Okay, but it is getting inhibitory impulses from globus pallidus externus. Globus pallidus externus. So when the cortex is stimulating the striatum, when the cortex is stimulating the striatum, then what will happen? This striatum will excite the external part of the globus pallidus, and it will release GABA as a neurotransmitter. So the GPE will be inhibited. So there will be disinhibition of the subthalamus, and it will stimulate this complex, and this will inhibit the thalamus, and thalamus won't generate signals to the striatum or the cortex. So the unwanted movement is inhibited. Now you can easily write about the functions of the basal ganglia. What is the first function? Controlling of the voluntary motor activity. So how it will control the voluntary motor activity? Through the loops. So what are those loops? Direct loop and indirect loop. Direct loop is having caudate loop and putamen loop. The caudate loop will control the cognitive control. Okay, and putamen loop will control the subconscious execution of some movements. Okay, this is a C for C. Cognitive control is by caudate loop. Putamen loop is involved in the subconscious execution of some movements, and it is also doing these loops are also doing timing and scaling of the intensity of movements. Okay, timing and scaling of the intensity of movements, and through the indirect loop also they are doing what they are doing, they are inhibiting the unwanted movement. And second one is the control of reflex muscular activity. Third one is the control of muscle tone via substantia nigra, because It is involving gamma motor neuron. So gamma motor neuron is under the control of substantia nigra. Through that, muscle tone will be controlled. And it has a role in arousal mechanism through globus pallidus. It is the main action, control of voluntary motor activity, and these three also the functions of the basal ganglia. What are they? Control of the reflex muscular activity, control of the muscle tone. So these two are responsible. These two are involving muscles. Control of reflex muscular activity and control of muscle tone, and it also having role in the arousal mechanism. That is the function of the that is another function of the globus pallidus. So we are not going into detail of these functions. We have discussed detail of the first function. Then the next video is about the disorders of the basal ganglia.